Hello friends, NCERT-Botany next episode. Plant growth and development, third one, dealing with hormones. So we can proceed to third episode of plant growth and development. So I will share the screen. I think screen came. So we can proceed. So today we'll be discussing in this episode 5 hormones. Theoretical aspect, usually 1 or 2 questions we will be getting for NEET exam and even theory exam. So first we can start with the discovery of PGR. So discovery of each of the 5 major groups of PGRs were serendipity. So what is meant by the term serendipity? Serendipity means happy accidental discovery. And in this case, it started with Charles Darwin and his son Francis Darwin observing in the coleoptile of a plant called canary grasses. Scientific name is Phalaris canarensis. That is how the discovery took place. After that, you can find a diagram. It is a famous bioassay test for auxin that we will be discussing it as we continue. Now, when we proceed to auxin further, in 1880, Charles Darwin and his son Francis Darwin found that the stimulus of unilateral illuminations were picked up by the coleoptile tip of Phalaris. Phalaris is canary grasses. But the bending response to unilateral light was produced at subapical part. So it will be like this. So from here, unilateral light comes touching on the tip portion. So the bending took place here, this part, not at the tip, that is on the subapical part, this way it bends. Now, auxin was the first plant hormone discovered, first isolated from samples of human urine. Now, after this, a series of experiments by different scientists conducted in this auxin. Some of them was all known need. See, when coleoptile is removed, no bending occurred because stimulus of light cannot be perceived because it is perceived by coleoptile tip. So no bending took place. That is the first experiment. I think first page is clear. Now we can proceed to second page. Second, when we start, when the coleoptile tip is covered with opaque means light will not pass through. So opaque material like tin foil, no bending took place. Third one, when coleoptile tip is covered with translucent caps, so light can pass through, bending took place. Fourth one, a scientist called A. Pal done experiments showing that with a special treatment, growth observed even in darkness. Then comes Boysen and Jensen. They observed that stimulus for bending can be transmitted to down through gelatin, but not through mica. That means light should be able to pass. Now, fifth one, his name is F.W. Wendt. He collected growth-stimulating substances in agar jelly. He observed that the hormone has got polar movement. Polar movement means tip to bottom. From this collected agar blocks, bending can be caused according to concentration. F.W. Wendt given the term auxin, connected with the tips of oat seedling coleoptile. Oat means Avina sativa. Now, sixth scientist, Kojal and et al. Isolated 3 chemicals from human urine, auxin A, auxin B, heteroauxin. These workers extracted auxins from urine concentrate at an acidic pH with ether. IAA is the most famous auxin. It is indole 3 acetic acid. So acidicity we can observe. Auxin is present in human urine, specifically persons suffering from pellagra. Pellagra is due to the deficiency of nicotinic acid or niacin. Auxin is acidic. Now, last scientist, Thiman. Thiman identified the chemical structure of auxin. So, roughly 7 scientists discuss, of which most famous is FWN, including giving the name auxin. Now comes precursor of auxin. It is tryptophan, an amino acid. Now comes classification of auxin. Different classifications available. 2 types we can just pass through. One is natural auxin, example IAA. One is synthetic auxin, example is NAA. Naphthalene acetic acid. 2,4-D means 2,4-dichlorophenoxacetic acid. That way the naming nomenclature. Second category is free auxin and bound auxin. Now comes physiological role, uses, applications of auxin. It is unlimited. All hormones, plenty of things will be there. Based on NCERT, will be proceeding just a few points extra, that's all. Otherwise, no meaning in discussing all the properties of auxin. So prevention of abscission. Abscission means falling off. Under normal level of auxin, no abscission or it will be suppressed. So for young parts, abscission not needed. For older parts, abscission is needed. So it is based on amount of auxin. I think this page also clear. Now we can proceed to next two. That is apical dominance. Usually, apical bud dominates over lateral or axillary buds. Or we can say apical buds will suppress lateral or axillary buds. It is mainly concerned with the auxin. And the diagram given there was a famous NCERT diagram. Tip is removed. So second diagram shows after removal of tip, lateral branches. Lateral branches means this one. This. Initially here. That will be lateral side growth takes place. So when apical bud is removed, that phenomenon is called decapitation. Lateral buds will develop. If again when we apply auxin at cut ends, lateral buds get suppressed. So this principle we can utilize for those plants which needs bushy nature and leaves commercially utilized. Third one, xylem, cell division and xylem differentiation. Auxin usually seen at tips, dividing areas, etc. Then fourth one, it is rooting. So auxin is otherwise called rooting hormone, mainly connected with the stem cuttings, and auxins used were NAA and IBA. IBA, it has got an exception because natural auxin, synthetic auxin, both way it has been classified. IBA means indole 3-butyric acid. Next is flowering. For flowering, we can utilize auxin, specifically NAA and 2,4-D. But the dilute solution and high concentrated solution have different effect in flowering. Dilute solution, flowering will be affected in pineapple and lychee. But high concentration will inhibit flowering in most of the plants. In such cases, when inhibition of flowering takes place, more leaves can be produced. So according to our need, flower or leaf, we can utilize these hormones. Now comes sixth point, parthenocarpy. So parthenocarpy means seedless fruits. So hormones used will be IAA, IBA, usually in low concentration, can produce parthenocarpic or seedless fruits, as in tomatoes. In certain natural fruits, we can find parthenocarpic. Example is banana. And scientists observe in such fruits, auxin amount is more. Then auxin can be used as a weedicide to nullify the weeds, specifically dicot weeds. Advantage is when we cultivate monocot plants or grassy area or lawns, LAWNS. So there should not be any dicot plants, so we can apply auxin. Dicots will be affected, monocots will be free of attack of auxin. Now comes, I think this page also clear. Agent Orange. It is a famous chemical used in Vietnam War to destroy orange cultivation. Agent Orange is a mixture of 2,4-D, 2,4,5-T. So many uses are there for each hormones. And after that, a totality has been given, so it will be easy for you to remember. This way you can make your own mnemonics. Bioassay, estimation, identification for auxin, it is by Avina curvature test. The first diagram you observe, it is curving. This is famous Avina curvature test. Root growth inhibition test. So these are some points in auxin. I think auxin is clear. Now we'll be proceeding to gibberellins. Gibberellins, when we discuss, Japanese scientists in Japan, they are more advanced in gibberellins because they know this hormone very early, at least a century ago. And initially, they observed rice seedling, the cultivation, grow excessively tall in their fields, and they become weak and sterile, and they will lose the balance and will be coming down. Name of this condition is called Bekane or foliage seedling disease. Kurosawa, the Japanese plant pathologist, 1926 itself identified the disease has been caused by a fungus, namely Gibberella fujikori. It is an ascomycetian fungus. It is a perfect fungus. It has got an imperfect partner. Imperfect fungus. It is Fusarium monaliforme. Wilder disease, Fusarium, connected with the bioclassification also. So perfect Gibberella fujikori, imperfect is Fusarium. Now Yabuta, another scientist, active substance was separated and named gibberellin. Kurosawa also found that the extract of fungus also caused appearance of disease symptoms in uninfected rice seedlings when it comes in contact. So disease can be spread. That is given by Kurosawa. Presently, more than 100 gibberellins identified, named as GA1, GA2, GA3. GA3 is most intensively studied gibberellin. GA3, that is itself is a question. And the ring system in gibberellin is called gibbane. Precursor is mevalonic acid, which is derived from acetyl coenzyme A. So some books it is written as acetyl coenzyme A, some book mevalonic acid. So mevalonic acid derived from acetyl coenzyme A. Now physiological effect and functions. One is bolting. It's an important item, bolting hormone, gibberellic acid. What is bolting? Increase in the internodal length due to inducing subapical meristem to develop faster. Beat. Another one, in case of rosette plants like cabbage and root crops like radish. Spraying gibberellins to sugarcane crop increase the length of the stem and can yield sugarcane directly means sugarcane as much as 20 tons per acre. Seed germination. So dormancy can be nullified by using gibberellic acid. So spraying will cause stimulation of mRNA, then hydrolytic enzyme, amylase, lipase, ribonuclease, and proteases. Gibberellins increase number and size of several fruits. Example is grapes. In apple fruits, size and shape is enhanced. Malting in barley grain. Malting. Overcoming dormancy of seed and bud. Delayed ripening, that is in citrus, and early maturity juvenile conifers. So many actions. I told you each hormone has got many actions. Now bioassay for gibberellic acid is dwarf mace test, P test, etc. Similarly, induction of alpha amylase in barley endosperm test. These are some bioassay for gibberellins. Now, as usual, you can find totality of gibberellins, including that bolting procedure. Then comes cytokinins. Name cytokinin is due to the phenomenon cytokinesis it performs, either alone or in association with auxin. Chemically, cytokinins are basic in nature, remaining four are more towards acidicity. So cytokinin is exception, it is basic. Discovery. Scooge and co-workers observed that tobacco callus proliferated only in addition with the auxin along with it provided either with the coconut milk or with the yeast extract DNA. So if we give auxin, no much proliferation, auxin should be accompanied or together with the coconut milk or yeast extract DNA that contains cytokinin. Now comes second scientist Miller et al. First cytokinin was discovered from degraded autoclaved herring sperm DNA. And the name is kinetin, N6-furfurylaminopurin. That itself was a question. Kinetin not naturally occurring in plants. Synthetic hormone. First natural cytokinin was obtained from unripened maize. Maize means chia grain. Then coconut milk, etc. Since it obtained from chia, the hormone is called zeatin. Many types of cytokinins are there. Some of them are constituents of tRNA. Which all places we can find cytokinin. Cytokines is so areas of cell division. Areas of cell division or roots, etc. Roots also a source of cytokinin synthesis. Chemically modified purines. It is basic. One is BAP, 6-benzylaminopurin. Other is IP, isopentenyl adenine. Now functions and physiological role. Again, I put in a circle so it will be easy for you. Cell division, cell elongation, morphogenesis. Now a term comes Richmond-Lang effect. So very famous. Richmond-Lang effect means delay of senescence done by cytokinins. Then increasing shelf life, tissue culture. All this area we can use cytokinins because cell division. Now in tissue culture, there is another important aspect. When auxin and cytokinin concentration are equals, we will be getting only callus. When auxin concentration is more than cytokinin concentration, we will be getting root. When auxin concentration less than cytokinin concentration, we will be getting shoot. So this type of action is called synergetic and antagonistic actions of hormones. And these three, auxin concentration equals cytokinin, auxin is more, auxin is less, what will happen is an important question. Sometimes people may not be knowing what is callus. So callus means an unorganized mass of cells. Richmond-Lang effect means delay of senescence in plants. So I think cytokinin is clear. Now we can proceed to next hormone. It is ethylene, one of the most commercially used hormone, ethylene. Ethylene is a gaseous plant hormone and more towards acidicity in nature. Ethylene can act as a promoter as well as inhibitor. So both ways it will work, but more towards inhibitor. Its discovery, cousins found that ripe oranges produced a volatile substance that hastened the ripening of unripened bananas nearby. Later, scientists identified that the volatile gas was ethylene. Which all area we can find ethylene? Almost all parts of the plant. Maximum synthesis at ripening of fruit and tissues undergoing senescence, so aging. Excess oxygen can also induce production of ethylene. So what it means? Many effects of excess oxygen are actually the effect produced by ethylene because excess oxygen can induce production of ethylene. It's a good area for question. Then a lot of features given. Gaseous, transverse growth, senescence, abscission, breaking of dormancy, growth of rice seedlings, leaves comes above the water, helps in root initiation, lateral roots, root hairs, fruit ripening, climatic fruit, fleshy, apple, mango, banana, flowering and fruit setting in pineapple, flowering in mango, sex expression towards femaleness, sprouting of storage organ, rhizome, comb, tuber, seeds, peanut, etc. Thinning of excess flowers and young fruits, cotton, cherry, walnut, elongation of stem in deep water rice plant. So all are important features. That is why given in a circle. Precursor of ethylene is methionine, and bioassay for ethylene is triple response test. Apical formation, horizontal swelling, that way it proceeds. Now, there is a liquid called ethaphone, and ability of that liquid is it can release gaseous ethylene in connection with the artificial ripening. And chemically, ethaphone is 2-chloroethyl phosphonic acid. All are important item for exam. I think ethylene is clear, most commercially used hormone. Now the last hormone for us is abscisic acid, commonly called stress hormone, ABA. It is acidic in nature, generally growth inhibitor. But remember, inhibition is only for the advantage of the plant, not against plant. Discovery is around mid-1960s, and different scientists reported different chemicals and names in connection with ABA. Some names include inhibitor beta, abscission 2, dormin. Later, it is identified all these different chemicals, what inhibitor B, abscission 2, dormin, all are chemically same. And a common name was given. It is called abscisic acid or ABA stress hormone. Occurrence is in active or dormant areas like bud seeds. Precursor is violosanthin. Some say it can be carotenoids. Now comes physiological effect and function. Abscission, dormancy of seeds and buds, inhibition of metabolism, leaf senescence, stoppage of transpiration, that is otherwise called antitranspirance. Ethylene can cause ABA can, not ethylene, ABA can cause complete closure of stomata and role in seed development. When condition is not favorable, seed should not develop. Now comes the last aspect, interaction among the PGR. Two-way hormones can interact. One is together, that is called synergetic. So for tissue culture, auxin, cytokinin. For root nodule formation, auxin and cytokinin. Sometimes hormones will be antagonistically acting. For example, ABA, that may cause seed dormancy. It can be broken by gibberellic acid. So ABA is otherwise called anti-gibberellic acid. So this way, the two hormones should work together. It is called synergetically. Otherwise, opposite manner, it is called antagonistically, and it is only for the beneficial nature of the plant. Now, 5 theory question as usual, 5 NEET model question with the key given. So this is the topic for us today. I think it is clear. So what all things we learned or passed through, starting with the hormones 5, discovery of hormones, Darwin, then auxin, their characters, features, classification of auxin, then physiological role, application. This way, each hormone, a lot of items are there. So basically, consider an NCERT, that is better. Then few extra things came for different exams. After auxin, we proceeded to gibberellic acid, features, then scientist, bolting, application. After that, we entered into cytokinin, exception, then features, scientist, chemical nature, then Richmond-Lang effect, tissue culture, how it operates. Then we entered into most commercially used hormone, that is ethylene, gaseous hormone. It can work as an inhibitor as well as promoter. Then its action, plenty of actions for ethylene, then precursor, then triple response test, ethaphone, etc. Then we entered into abscisic acid, stress hormone, features, then antagonistic and synergetic action. After that, 5 theory question, 5 NEET model question. This is the topic for today's episode. So by which plant growth and development, all areas are over. So we'll be meeting later with a new topic. So we can see later. If you like the episodes, subscribe or like it, share it, comment on it. We can meet later. Thank you.